Let's see if it works. It should be work this time. Now it's in Michigan, it's springtime. There's no snows. And the weather is pretty good. It's sunny, shiny. It's very nice. Okay. So there's no. So let me see. Okay. Okay, today is the third lecture, and this lecture is about the structure and function relationship of the channelrhodopsin. So generally say, in this lecture, we will go through with the 3D structure of the channelrhodopsin, this protein, and then we're talking about why we need to know this structure and what can we do with the research result discoveries of this structure. Generally say the overall structure of the channel, it has 3 cavities. It has an extracellular cavity, central constructional site, and this site is located with the Schiffer base of the ultrasound retina. So this site can be changed by after catch the photon by the ultrasound retina. Then the conformation change of the retina can change this central constructional sites and amino acid residues relationship, like forming hydrogen bond, break up old bond, form new bond, then make this channel to be open. And another part cavity is the intracellular cavity. So based on the 2D, the structure analysis of the amino acid of channelrhodopsin, we know that it has a 7 transmembrane domain, and all transretinal is binding in the center of the whole molecules. Channelrhodopsin is a channel, so when you catch the light, it itself can cause, like for the channelrhodopsin 2, CHR2, it can cause the cations flux into the cell and depolarize the cell. But for human rhodopsin, it's a G protein coupled receptor. And after activation, it will activate a downstream G protein. It's showing like this. That's a human rhodopsin. But so the whole rhodopsin family, it was including like, for example, the chloride pump, the rhodopsin, and this is a proton pump that pumping out that bacterial rhodopsin. And as other like a sodium pump and the chloride channels, and the channelrhodopsin, the cation channels that can cause the cations to flux into the cell, and the proton pump that can depolarize the cell, and also the rhodopsin linked to PDE, phosphorescerase. So this all of these proteins are included in the whole the family of the rhodopsin family. Of course, also including the human rhodopsin that's a G protein coupled receptor. And the following lecture, we're going to talking about the channelrhodopsin. That's because, like for the rhodopsin, the chloride pump or the proton pump, the bacterial rhodopsin, these pumps require higher protein expression level and stronger light stimulation to cause a very significant membrane potential change. But the channelrhodopsin as a channel, it's the ion like transmembrane is much huge than the pumps. So, and the scientists are most focused on channelrhodopsin, and the first structure research is focused on channelrhodopsin. So this work is published in Nature in at 2012 from a Japanese group, Kato. And in their study, they discovered that the channelrhodopsin, it forms a dimer. Let's see, I will try this, do an annotation spotlight, this one here. So it forms a dimer. That's because in the extracellular part of the N-terminal, there's a three cysteine residue there. So it can forms a bond that links the two monomer channelrhodopsin together to form a dimer. But the channels are located within each monomer. So here there's a channel, here there's another channel. So the ion flux is between these four transmembrane domains, the transmembrane domain 1, 2, 3, and 7. And this magenta color is labeled ultraretina binding in the center of the channelrhodopsin and forms a Schiffer base with the channelrhodopsin proteins. In the N-terminal is at the extracellular, C-terminal in the intracellular, and the ion conductance in the center of the 4 alpha helix. So in this 2012 research article, actually is not to discover the structure of either channelrhodopsin 1 or channelrhodopsin 2. I think maybe that's due to easier to form the structure, or like for experimental advantage, they produce the chimera that forms that 1 to 5 alpha helix, and N-terminal is coming from CHR1, and the last two alpha helix and C-terminal is coming from CHR2. So it's a chimera. It's either not CHR1 nor CHR2. But when we focus to take a look at the sequence, and we can find that actually from the transmembrane 3, actually it's identical. And for the transmembrane 4, it's also quite similar, not big difference. Geometry is the same. So we can say that this C1, C2 chimera is like the first 5 is from the channel CHR1, but actually from the third alpha helix, the transmembrane domain, is quite similar to the channelrhodopsin 2. Now, I put all of the amino acids in the figure, and this 3 site, the C labeled with C, that's a cysteine, is a cysteine residue. That is the place that can form a dimer with another channelrhodopsin monomer. And that's the oligomerization that from the bond, from the cysteine to cysteine bond of the N-terminal. And the most important and catch people's eye for those ion selectivity and ion permeability, that's in the center of the transmembrane domain number 2. That's the E19 and E97 and the cysteine 128. And it's believed that this three site is very critical for the channel's ion selectivity that he choose to let the cations to efflux into. And later we will know the modern and genetic engineering that modified those sites can totally change the characteristic of the channel. And here it shows that this ion influx to the pore site is between the transmembrane domain 1, 2, 3, and 7. And here it shows the ion flows pathway. And the lower side this shows an extracellular, and this is the intracellular. So at the resting state, when the closed state, this more close to the intracellular part is closed by several hydrogen bond, and the pore that the opening that is close to the extracellular part is opening, and the critical site is here is determined by the E136 and E140, and this site, these two amino acid, the E stand for the glutamate, and this is critical for ion selectivity and the conductance and determine the kinetic of the channelrhodopsin. And it may also determine the action spectrum. But this is the whole structure result of the channelrhodopsin. The actual site is always opening, and the critical sites that the E136 and the E140 is located here. And after the transformation change of the all-trans retina will cause this structure change, and different bonds will be formed, and this pathway will be opened, and the ions can flux into. So this two site is labeled here. So it's on the second transmembrane domain. That's for the channelrhodopsin 2. That's E97 and E100. So generally say the architecture of channelrhodopsin's ion conducting pathway is including the extracellular cavity and the central critical point, the constriction site, and the intracellular cavity. So both of here's a cavity, but only after channels go opening, then this cavity can be linked together, then to form a channel. And this is at a closed state. We can notice that there's two parts of the extracellular cavity, one larger and one is smaller. And it's known that when the channel conformation change, when the channels are open, and these two cavity will be linked, also be linked together to form an even larger opening to open to face towards the extracellular part. And the extracellular cavity is generally larger than the intracellular cavity. Then why is it so important to know the molecular structure of channelrhodopsin? That's because if we know it, we know the detail of it, we can change it. It's like if we know the detail of the car, then if the car is broken, we can fix it. And if we know the blueprint of a car, then we can make it. So after 2 years after the structure results is been published, then in 2014 in Science paper, that's from And this is from Deser's group. And they made a 9-point mutation. 9-point mutations to this channelrhodopsin structure, and they can change this cation channel to ion channel. That means that they can pass through chloride. So at the same year, in another group, in Heckman's group, they do a very clever modification. They find in another site of the channelrhodopsin, the E19, changed to lysine or arginine. Let's change the glutamate residue to lysine and arginine amino acid residue that changed from a negative charged residue to positive charge. So at this site, previously this negative charge residue were binding to like cations like calcium or sodium, but after this modification, then you were binding to like a negative charged ion like a chloride. So then this function of the channel is totally switched from passing through cation to passing through like chloride, a negative charged ion. Of course, in this work, they do not only change the one amino acid, actually they changed the 2 or more. But these 2 sites are more critical for the function of the channelrhodopsin. So in this chapter, and the author also lists all of the structure that is widely used, nature finding the channelrhodopsin variant, or widely used engineered channelrhodopsin variant like RICH-R, like RC1C2. Thank you.